speaker is Beverly Rzigalinski, who has a BS in biology from Rutgers University in New Jersey, MS in biochemistry and toxicology from New York University, and a PhD in biochemistry and pharmacology from Eastern Virginia Medical School and the Old Dominion University. Dr. Rzigalinski is currently the professor of pharmacology at the Virginia College of Osteopathic Medicine and also has appointments at Virginia Tech, Wake Forest School of Biomedical Engineering, and the Virginia Maryland College of Veterinary Medicine. We look forward to hear from Dr. Rzigalinski. That's the actuator. Green turns forward, and there's the laser.
Okay. All right, I'm set. So I'm going to speak to you today from a different side, more from the biomedical side of things, particularly with regard to nanotechnology and treatment of diseases that are associated with oxidative stress. To a biomedical scientist, of course, oxidative stress is caused by generation of what we call free radical, simply a compound with an unpaired electron that will go about within the cell and wreak havoc on cellular macromolecules such as lipids, proteins, DNA, and it will actually do a lot of damage in that vein. That being said, what we've started to work with is a compound that Jim mentioned called cerium oxide, actually a nano compound, if you will. Cerium, of course, is a rare earth element. You all know that. I'm with a bunch of engineers here. It's like I've kind of found myself in a very strange place as things went on, whereas I was a neuroscientist, and then I started dealing with engineers, and the biochemists think I'm nuts, so I'm like really stuck in the middle now. But that's okay. It's good here. Cerium atoms surrounded by an oxide lattice are what comprise this nanoparticle. And of course, as Jim said, the cerium has multiple valence states, +3 and +4. And the oxide lattice has what you folks call numerous defects, or electron holes in the lattice. And as you probably all really well know, the micro-sized particles are used in catalytic converters and for other industrial and materials-related redox reactions. Now, of course, at the nanoscale, we think that the redox activity of this material is even more potent, shown here with the cerium atoms and, of course, the oxide lattice. And, of course, these particles behave a little bit differently from their micro-sized counterparts. We don't see much biological activity of the nature that I'm going to talk about with the micro-sized counterparts or with the salts of cerium or the chlorides of cerium, but we do see what I'm about to show you with the nanoparticles. Now, about 7 years ago, just kind of by happenstance, we found that cerium oxide could extend the lifespan of mixed brain cells in culture by a tremendous amount. Our average lifespan of mixed brain cells, and this is essentially what we call an organotypic brain cell culture. It's basically a brain in a dish. We take it out of a neonatal rat, so the culture itself has neurons, astrocytes, microglia, the spectrum of cells that are in a normal brain. And they will survive in vitro for about 21 to 28 days. They die out naturally. The cultures undergo a natural cell death. Most people think that this is due to oxidative stress. Cells in culture, when you take them out of an animal, almost unilaterally have a much higher degree of oxidative stress than when they are in the body. So that's why everyone kind of thinks that they die out at this age. So the cerium oxide treatment extended it dramatically. We've actually got some real-time videos of these cells signaling on our website. Most we've seen is about 123 days, anywhere between 60 and 120. Now, I'll point out that we only treat the cells once. They are not continually treated with the nanoparticles. They're treated once on day 10 in vitro. So this lasts them throughout the extended lifespan. And this is what they kind of look like. This is a 27-day in vitro organotypic cell culture right here. And as you can see, there's not much left in it. This might look like a neuron. It's probably an oligodendrocyte. This is basically the blank tissue culture well. Here we have our 27-day in vitro Syria treated culture. And again, this is one treatment on day 10. And you'll see that in the background, there's a very robust astrocyte layer. And here we have some nice pyramidal neurons here, a lot of axons. And at 123 days in vitro, we see a similar scenario. Nice, robust astrocyte background and a nice neuronal upper layer. The astrocytes kind of stay in the background of the tissue culture plate. The neurons grow on top of them, so the two cells are very closely linked. And these are cortical cultures. Now, it's wonderful if you can keep cells alive longer. That's a wonderful thing. But when it comes to aging, you want to maintain function as well. And for that, what we've looked at is the signaling capacity of these neurons in these mixed organotypic cultures as the cultures age. We look at intracellular free calcium using microspectrophotometry, and these are a few sample tracings. Now, mind you, in a typical field of cells, there's probably about 50 or 60 neurons. We analyze all of them. This is just a sample tracing of what we see. Now, in a normal culture, that's about maybe 18 or 19 days old. You'll see what we see right here, and I haven't put the control on here to keep the slide from looking a little bit too busy. But you see what we call basal levels of calcium oscillations. So the cells, the neurons there are basically talking to one another very robustly without any further stimulation. And of course, we stimulate with glutamate. We see a nice, robust response in glutamate. Why glutamate? That's our major cortical neurotransmitter. Now, I'll draw your attention to this red line up here. This is a neuron in a 28-day-old control. Notice the basal calcium is high. This is typical of something of a cell that's going on to die. Basal calcium is high. There's no oscillations. They're not really talking to each other very much. And when we stimulate with glutamate, there's no response. So this is a typical response of a neuron that's on its way out. Here we have our 68-day-old nano-treated and 28-day-old nano-treated. Very nice, robust calcium signaling, nice response to glutamate in a return to basal. So they are indeed preserving function with their extended age. So we began to question what was going on. And what we feel is happening is, as Jim had mentioned, cerium can flip-flop between a +3 and the +4 state. This is a typical redox reaction, though. So along will come a free radical. Cerium +3 will flip to +4. We know there's some kind of change in the oxide lattice here. Because what we also feel is going on is that the ability of these nanoparticles to scavenge a free radical is regenerative. We're giving one dose on day 10. Okay, that's lasting for the lifespan of these cells, and I'll show you some other information on this in a little bit. And what we think is there is some kind of reaction with perhaps water, hydroxyl ions, or hydrogen that is absorbed to the surface that causes it to revert to the +3 state. Now, we originally thought that the +3 to +4 ratio of the cerium was the important thing here. So we've gone on to do a lot of work comparing different cerium oxide nanoparticles with different amounts of +3, different amounts of +4, different faces. And as I understand it, you engineering folks refer to it as cerium 100 versus cerium 110, different faces of the crystal lattice. Thinking that, well, this must be, you know, the more cerium +3 we have, the better an antioxidant this is going to be. Wrong. So we think the oxide lattice is a heavy contributor here, and we don't understand why this is happening at the biological level. That's why we heavily count on our interactions with the engineering groups for guidance here and for manufacturing many different types of nanoparticles. Now, for the rest of this talk, you're going to hear me speaking about 10 nanometer seria and 7 nanometer seria. These are just two sizes we've compared. And one of the biggest things that we noticed about 5 years ago was we said, hey guys, you need to give us nanoparticles that are the same. When you make them in your lab, from batch to batch, they really need to be the same. And although our engineering group still makes some of these, we have convinced two Nanophase Incorporated in Romeoville, Illinois, is now making a pharmaceutical-grade 10 nanometer seria that's open to the research community, and Advanced Powder Technologies in Australia is making a standard 7, which we characterize and look at for purity, toxicology, et cetera. Because pharmaceutically, when you're going to move this stuff up to the biomedical field, you need to be consistent from batch to batch so that we can make the strides that we need to make. So that's just one reminder about the way we have to look at things when we're actually moving this from the benchtop or from your engineering benchtop to bedside. We need to be really consistent, really important. Anyway, I'm deviating. Here we're looking at, does Seria really scavenge free radicals? So we're looking at electron paramagnetic resonance spectroscopy. We're generating the Fenton reaction in an EPR tube, and we see a characteristic 4-line spectrum for the hydroxyl radical. We add 10 nanometer seria, it nicely quenches it, gets rid of that hydroxyl radical. 7 nanometer, same thing. Iron-doped seria, I'm not going to get into. We're actually looking at iron-doped for some people that work with spions to see if cerium can perhaps decrease radical production of the iron nanoparticles. Now, biologically, superoxide is also a very important radical. So we generated that by irradiating riboflavin, looking at the EPR spectra. And again, we see some quenching by the 7 nanometer seria, but more by the 10. Now, why should this be? 7 has more surface area than 10. We really should see more quenching here than here. Okay? So this means we still have quite a bit more work to do here. Just because I'm saying it's 7 and this one is 10, yeah, 7 has more surface area, but what's the surface structure? This is another thing that we really haven't considered that probably at the biomedical level has a lot to do with it. How many dips, irregularities? We think of a crystal as having, and this is a crystal, having a solid face, but it doesn't look like that at all. It's probably very crinated. Are there more cavities in one than the other? So we think that that may also be having something to do here. So probably not just size, but shape also. And we also went on to say, okay, if it's acting like an antioxidant, that when we put it in a typical biomedical assay for an enzyme, does it have antioxidant activity? And these experiments are really relatively simple. We just took some standard off-the-shelf kit assays for enzymes that scavenge free radicals. One being catalase that scavenges hydrogen peroxide, SOD that gets rid of superoxide, and glutathione peroxidase, which maintains a natural antioxidant system in the body. And again, we're looking at our 10 nanometer and our 7 nanometer particles, and all of these particles have activity that is equivalent to the enzyme. So although this is a nanoparticle, it acts like catalase, and it acts like SOD, and it acts like glutathione peroxidase. So it has an activity that is very similar to our natural antioxidant scavenging activities in the body. And once again, we see the 7s being a little bit more, the 10s being a little bit more active than the 7s here. So the particles actually act in a manner very similar to our own enzymatic systems. We went on to look at some UV-mediated oxidative stress. Ultraviolet radiation is a good way to simulate oxidative stress in the dish. And we compared our cerium oxide, and again, these are all one dose delivered on day 10, to vitamin E, acetylcysteine, and melatonin. And here's our control. 5 minutes of UV. Draw your attention to the yellow bar here, which is the cerium. Here, we're looking at injured cells through uptake of propydium iodide. That will show us dead or damaged cells. So we see that cerium affords us some very good protection as compared to no treatment. It also appears to be better than the vitamin E, the acetylcysteine, but look at this one, melatonin, 5 minutes. It's about equivalent to it. Where we do see differences is with the long term. When you increase your radical insult, here to 15 minutes, serum is the only one that gives us some nice extended protection. And this again goes back to the fact that these pharmacological ones, vitamin E, acetylcysteine, melatonin, one free radical, one molecule of it goes. In the case of cerium, it can handle multiple free radicals. Okay, so that's a very, very important point when we look at the pharmacology here. And we also looked at some H2O2, hydrogen peroxide mediated injury. Now in this one, we're comparing 3 different sizes, 50s, 10s, and 7s. Here is our control, which is hydrogen peroxide injury, right, which kills quite a bit of the cells, almost 100%, killing at the dose we're using here. So if we look at the blue bars, we see no protection from the 50 nanometer particles. Our EM studies suggest that they don't really get into the cell very well. It's got to be under about 20 to get into the, at least neuronal cells and endothelial cells. Now I'll draw your attention to the purple. Here's our 10s. Nice protection. This is dose on the bottom here. Nice protection, but if you look at the dose response curve, it's bell-shaped. Somewhere between 10 and 100 nanometers is our optimum dose. At a lower dose, no effects, and at a higher dose, no effects. Many pharmacological agents are bell-shaped, do have bell-shaped curves, but what we really have to realize when we're thinking about these nanoparticles and oxidative stress is we now know that free radicals are part of normal signal transduction in the cell. Growth factor receptors, calcium channels, the endoplasmic reticulum relies on hydroxyl radicals and superoxide radicals and nitroxyl radicals for normal signaling. Now, we knew this about 10 or 20 years ago because NO, of course, is used for vasodilation and neuronal signaling. But the plot has even thickened more, such that we believe that there is a basal level of free radical production necessary. When we see these effects starting to, the beneficial effects rather, starting to go away, we may be approaching that level of cerium in the cell that's going to impart negative effects. So we need to identify these things, be very careful what we're looking at. Now, we've moved up to looking at some biomedical treatments for common diseases, first being brain injury. Before I did a lot of nanotechnology, I thought I was going to step backwards off of this. Before I got into nanotechnology, I did a lot of work with traumatic brain injury. And for this, we use an in vitro model for TBI. I won't go into the fine points of the model, but we pre-treated ourselves with cerium oxide nanoparticles once, again, day 10 in vitro, and went on to injure them with what one would consider a moderate level of head injury and a severe, and these are, again, cortical cultures. And we see some wonderful neuroprotection from the cerium oxide nanoparticles. Now, you're never going to be able to pre-treat an individual for a brain injury. It's just not going to happen. So we looked at post-injury treatment, and we see we still get significant protection when we deliver these particles post-injury. We can actually go up to 3 hours post-injury and still see nice protection. Now, our group has previously shown in some earlier studies that free radical production is a component of traumatic brain injury. So this is how we feel that it is acting here. But I'll also note, and I don't really have time to get into it in this talk, they're also very potent anti-inflammatory agents, and these are mixed brain cell cultures, so there are microglia in here, and they do blunt the inflammatory response of microglia. Now, one of our other interests, particularly with TBI, is Are the neurons still functional once again? And if we look at this group of 3 bars, here we're looking at the percentage of neurons that are examined. And down here, we've grouped them into 3 levels. In a normal neuronal culture, these cells respond to glutamate, which is their major neurotransmitter, mostly within a certain range of a response, a change in calcium. There is a small proportion of what we call hyperresponders and a small proportion of hyporesponders. After our simulated traumatic brain injury, that proportion changes. There are more hypo and hyper responders, and the normal responders goes down. This is very consistent with what's seen in human and animal experiments. In our cerium-treated cells, we see that the normal distribution, compare this set of green ones to this set of yellow ones, is more preserved when the cells are treated with cerium, suggesting that at least for this time period, okay, we see improvement with cerium. Cerium can maintain the function. Now, we've moved up to looking at some Alzheimer's disease models. Alan Butterfield's group in the University of Kentucky has shown that when amyloid beta aggregates, it generates free radicals. And we've been able to reproduce that up here. This is amyloid beta peptide aggregating in a culture dish, and we get a characteristic 6-line EPR spectrum that we can block very nicely with cerium. And of course, these are 2 controls here showing that cerium alone does not generate any free radicals. Well, once we saw that, we said, okay, what does it do with the neuronal death model of Alzheimer's disease? And in this case, we treated pure neurons. Now we've switched cultures. We're working now in pure neurons only. And we gave them amyloid beta peptide, which does kill pure neurons through several different mechanisms that I won't go into, but here is the cell death associated with amyloid beta. And we see we can block that extremely nicely with 10 and 100 nanomolar cerium oxide. And this is the 10 nanometer size. We haven't really done a size profile on this one. To give you a little bit more of an idea of what it looks like, this is a control neuronal culture. It looks like a tangled web of neurons. They're all oscillating in terms of intracellular free calcium. There's a lot of nice dark processes. This is our amyloid beta treated culture. What I want to draw your attention to, you see the processes here. Here they are fragmenting. They're beating up into little small beads. Now here is our 10 nanomolar of 10 nanometer cerium and 100 nanomolar right here. You can see we have some very nice processes that are maintained. And when we go to look at signaling, we see that the spontaneous calcium oscillations are maintained very nicely in these amyloid beta treated cultures. So it appears to be working very well. And that actual project is going to be moving up to a Drosophila model of Alzheimer's disease at the end of this month. Speaking of Drosophila, this is our next step up. Many of the aging communities use Drosophila as a model of aging, so I'll quickly go through a couple of these and also Parkinson's disease. We looked at feeding ceria to Drosophila in the fly food, okay? And that's shown on this graph. I'll draw your attention to the white line first. These are the controls, okay? These are controls as they age. This is percent survival here, typical mortality curve. This is a cohort of 100 flies. And here's our cerium-treated cells right here. They live the extension in median lifespan is about 32%. Maximal lifespan, which is the average lifespan of the last 10% of the survivors, last 10% cohort, is increased by about 26%. Now, what I'll also point out, if you look at this dotted line, this is what we call negative geotaxis, a measurement of function. When fruit flies grow in a long, tall vial, when you tap that vial, all the flies go to the bottom. Their natural response is to climb back up. That's called negative geotaxis, and that response goes away as they age. And you could see that in the control, and it's preserved for a longer period in the nano-treated. We've also been able to show that The nanoparticles are very protective in a Parkinson's disease model of the fly that involves paraquat, and you can just glance through this quickly by comparing the green bars to the blue bars. The greens survive a lot longer. So this one is now moving up to an animal model of Parkinson's disease. We've got our start in animal models, mostly toxicological. We are injecting them. We've gone up to 150 micrograms total. Okay? No toxicity, no abnormal pathology yet, but I really wouldn't be convinced on that. To give you a summary, this looks like a very promising future nanopharmaceutical. We've got a lot more ground to cover. The main question we need to answer is how much free radical scavenging is too much? That's going to be very important. And ask me about this later, but that longevity curve I showed you, the reason I had females up there, sorry, guys, doesn't work very well in the males. We think you could always say, you know, it's the better half that's going to live longer, so that's okay. But we think that's a dose thing. And you can ask me about that later. We think that's another one of too much free radical scavenging. So we need to control it. And the toxicity profiles on these are really going to be critical. And I'll get everybody's name up. This is the lab and all of our collaborators, and I'll stop here.